Hello everybody, my name is Elizabeth and I work at the University of Queensland, and specifically I work in an institution called QAFI. And QAFI focuses on agricultural and food improvement research. And I sit within the Centre for Animal Science, and most of my research focuses on improving livestock production. And up front I just wanted to acknowledge that the examples that I'm going to give you today of my work are really a team effort. All of these people that stand up in front of you and talk about the work they're doing, none of them did it alone. So I want to acknowledge the team up front. So today I'm going to be talking about revolutionizing agriculture by harnessing the power of nanopore sequencing. So a lot of you are here today telling us about how we can use nanopore sequencing in a clinical setting, how we can use it to improve human health. But there's a bigger problem coming for us. Projections show that the world population by 2050 is going to hit almost 10 billion people. 10 billion people on Earth. And the problem with that is that we are already struggling to feed the people that live on Earth today. How are we going to feed the extra people when there is 10 billion of us crowded in here? Not only that, but we have a finite set of resources. The world that we populate, it's not growing. But additionally, as these 10 billion people come into existence and need somewhere to live, our cities grow. And our cities encroach on our best agricultural land. So not only do we have more people to feed, we have less room to do it in. So we have to do more with less. We have to produce more food, more nutrition for these people so that they can live happy, healthy lives and do more nanopore sequencing. But we have to do it with less. How are we going to do that? Well, I think part of the solution is genomics. One of the ways that we can use genomics to improve agricultural production is something called genomic breeding. Now, a lot of you have heard this term polygenic risk score. And people like to throw that around, but they often don't really explain what it is. It's something that started in agriculture. And it started in agriculture to determine the genetic merit of individual organisms. It was first implemented in dairy cattle, and it is used worldwide now. Basically, the premise is this. You take markers across the entire genome, you calculate the effect that they have on your trait of interest, and then when you genotype new individuals, you can just apply that prediction algorithm to get an idea of their genetic value. So that means that instead of waiting for those animals to grow for 6 years till they start having calves and producing milk, for example, or they reach their full size to get an idea of how well they're going to perform, you can do it the day they are born. You can take a piece of DNA, you can genotype that DNA, and you can know, what do you want to do with that animal? Do you want to keep it? Do you want to breed from it? Is it worthwhile for you? And we can do this for all kinds of traits. We can do it for growth rate, we can do it for longevity, we can do it for fertility metrics, things that it's impossible to measure on live animals. And we can also do it to methane. And I'm going to come back to the methane story later on. But I wanted to talk to you today about two separate projects that my team is working on. One is looking at decentralizing genomic information. And I think that we always say that when people copy what we do, that's the highest form of praise. So thank you, Nanopore, for copying what we've been doing. And the other one is saving the world one cow at a time. And that's where the methane story comes in. Now, this is one of my PhD students, Harry Lamb. And he has just finished his PhD and just graduated. And his work has been looking at using nanopore sequencing to perform genomic predictions for genomic breeding. So the actual prediction, genomic predictions, that's when we're predicting the merit of that animal. So you'll find I may switch between those terms a little bit. But Harry had a dream. And this was basically his dream. Currently, if you want to perform genomic prediction on a cow, you will take an ear sample or pull out some tail hair, and you will pop that in the post, and you will send it away to a commercial sequencing provider. So it spends about 2 weeks in the post trying to get there. And then it gets to the lab, and then it kind of sits on the bench for a while. And they finally open the tube, and they genotype them. And they send you the results back a couple of weeks later. So you're looking at a 6 to 8 week turnaround. Now, I don't know about you, but cows are pretty big. People don't like rounding them up and bringing them in a heap of times, right? So that's really inconvenient. And it's really inconvenient in Northern Australia where people only see their animals once a year because you're not going to hold them in the yards for 8 weeks. So what if we could use nanopore sequencing to take a sample, sequence it, and within 24 hours, while the cows are still in the yards, return our breeding values and make our management decisions? Well, we needed to know if we did that, would we be making the same decisions as what we would do with snip arrays? So what Harry did was he took some samples that had snip array values. So we had a HD snip array, 700,000 markers, and an LD snip array, 35,000 markers. And then he sequenced those same samples, those same animals, using nanopore sequencing from 0.05 to 2-fold coverage. And then he calculated those breeding values. And when he calculated the breeding values for all of the different depths, what he found was that if you sequence an animal to 0.5-fold coverage, you can get the same genomic breeding values as if you had genotyped them on the high-density snip array. If you sequence those animals to 0.1-fold coverage, you can get the same breeding values as you would get on the low-density snip array, which is the industry standard. So that was great. And this is one example, body weight in this case. But we wanted to test it on different traits and on different cattle. So again, Harry went out and he went to his mate's farm and he took some ear punches from his friend's cattle. And he wanted to look at a host of different important traits. He wanted to look at body condition score, he wanted to look at height, he wanted to look at weight, and very importantly, he wanted to look at fertility, because fertility is a driver of profitability, and fertility is a driver of low methane emissions. So, how did he go? Well, he took these animals and he sent a sample away for sequencing at one of the large multinational SNP sequencing providers, and he did his 0.1-fold coverage sequencing on them as well. And again, what he found was that the breeding values that he calculated were effectively the same, exactly as he had predicted from the previous study. The other thing that he wanted to look at is bias, because we don't want to be using nanopore sequencing and introducing systematic bias into our results, right? Because every technology has a systematic bias, and we want to make sure that we're not giving the farmers artificially high or low values. And so that's what you can see there on the second plot. The dotted line is a perfect match between the SNP array genotyping and the nanopore-based genotyping. And the solid black line is the correlation that he got. And you can see they sit on top of each other. That means that there's no systematic bias, whether you're making breeding values using a SNP array or whether you're making breeding values using low-pass nanopore sequencing. But most producers, most people, most farmers, they don't really care about the numbers. The numbers on the page don't matter. What matters is what is the decision they're going to make with those numbers. And most people, when they do this sort of thing, they're looking for which animals do I keep to breed from? Which bulls do I want to be the father of the next generation of cattle? So what he did was he grouped them into bands based on the breeding values that he got from the SNP array and from the nanopore sequencing. And he asked the question, do these animals re-rank? In other words, would any given producer make a different decision based on the nanopore data than the SNP array data? And the answer was that there was almost no re-ranking. The decision the producers would make based on those values were almost identical. There was one animal that switched from the second band to the top band, and vice versa. The rest were all identical. So that worked really well. So not only is the correlation very high, but the decisions producers would make with this data is near on identical. So that's great. We can go out and we can use nanopore sequencing to genotype animals, and we can do that now. But there's another problem facing us. And that problem is climate change. And that problem is an issue because I live on this earth and I like it. And I would like it to still be here for me to live on. So this is a problem we've got to deal with. Now, climate change in the cattle industry, it's a funny problem because cattle, they don't produce as well when they're really hot or when there's severe weather events. So one issue is that they just actually produce less food to feed those 10 billion people that are coming. The other issue is that they directly contribute to climate change. And that's something that as an industry we have to deal with. So, if you imagine a cow, it's very big, and you think, oh, so cute, love it. But what you're not seeing is that cow is actually a massive fermentation vat. So a cow will eat some grass, and you think they're going to digest the grass, but the cow does not digest the grass. The cow has a massive chamber in its stomach called the rumen, and that rumen is full of microbes. And it is those microbes that digest the grass. That's why cows can live a happy life just eating grass, and we can't do it. They can change low-quality vegetative nutrients into high-quality protein. We can't do that. And they can do it because of these microbes in the rumen. And that's great, except that as a byproduct of that fermentation process, methane is produced. And methane is a strong greenhouse gas, 20 times stronger than carbon dioxide. And these microbes in the rumen called methanogens, they produce this methane and the cows burp it out. That's the cow burp thing that you've all heard about. But the one thing we have going for us is there is massive variation from animal to animal in how much methane they actually produce. And about 10 years ago, I showed that you can use the rumen microbiome, you can use that population of microbes in the rumen to identify high and low methane emitting cattle. But when I did that back then, we were using Illumina data, so we were using short read data. And the problem with short read data is that when you are trying to look at a microbiome, such a tiny percentage of that data is actually classifiable. You can only classify around 6% of the data if you use short read sequencing. So of course, because we have a P24, we wanted to change that over to nanopore sequencing. But we wanted to understand what are the things that are actually going to affect the population that we observe when we use nanopore sequencing. So this work was done by a master's student of mine, Ziming Chen, and he wanted to look at what are the different factors in nanopore sequencing that are going to affect how usable that data is for predicting methane. So what he did was he took a single rumen sample and he split it up into many, many technical replicates. So the actual population was the same, but just technical replicates that were treated differently. And he looked at using different base calling algorithms, different sequencing kits, and different DNA extraction methods. And overall, the biggest thing that affected how usable those reads were just the read length. That's it. If you get longer reads, they're more classifiable, you get more usable data out of what you sequence. The other thing he saw was that if you use the higher quality base calling algorithms, you can get up to a 10% bump in the classifiable data. So instead of only being able to use 6% of our data on the short read systems, now suddenly we can use up to 60% of our data. And that means we have to produce a lot less data. And of course, data management is kind of annoying, so that's great. Really happy with that. So after that, we're pretty confident that going forward, we can use nanopore sequencing of the microbiome to identify low methane emission cattle. We have the models working, we have the lab work working, we know that we can do it. So this means that not only can we go out and we can genotype cattle on the nanopore system, we can also identify which of those cattle are going to produce lower methane emissions, which means that we can go out and we can specifically breed for more environmentally friendly cattle that grow better using the nanopore system. So finally, we're heading down a path where we can produce more food, more sustainably, with less. Now, I just wanted to thank the funders of this work, Meat and Livestock Australia, who have a large program focused on reduction of emissions from the red meat sector, the University of Queensland, who got behind buying the nanopore system for us to do all of this amazing work on, and the team, especially Harry, Luan, Ziming and Shen, and also Nanopore for this opportunity to share this work with you. Thank you so much for listening.